Hi, I'm Ariella Golbin, and my project focused on characterizing the role of the mitogen activated protein kinase 7, or MKK7 gene, in developing salt tolerance. Soil salinization has become increasingly prevalent in various parts of the world. Soil salinization is characterized by having a solute concentration of greater than 4 decisiemens per meter. Currently, 7% of all agricultural land is salinized. This is projected to increase to 50% by 2050. Plants exposed to salinity will experience inhibition of growth as well as decreased yield. Ion imbalance and osmotic imbalance result from sodium ion accumulation, leading to an imbalance of water in the cells of plants. Due to the accumulation of reactive oxygen species, or ROS, in response to salt stress, several metabolic pathways can be damaged. To combat this, plants have many mechanisms. This includes the production of antioxidants, the activation of transcription factors that induce stress responses, the activation of signaling molecules, ion channel and transporter activation in order to balance sodium ion concentration, and the production of osmolytes that maintain membrane stability. Salt can cause a change in many signal transduction pathways, such as the MAP kinase pathway, which is conserved in plants and animals, and is responsible for transducing extracellular signals to intracellular responses. This is in order to regulate plant cell survival in response to stress. The effect of salinity on a plant, as well as the response of a plant to salt, is based on many factors. Different plant species have different tolerance and responses, depending on the dosage of salt and period of exposure. For example, salt can be added gradually to the soil, or it can happen immediately in large quantities. This is referred to as salt stress versus salt shock. Salt stress is characterized by a gradual change in gene expression as well as ionic stress that leads to osmotic stress. Salt shock, however, causes a sudden change in gene expression as well as osmotic shock that can lead to cell plasmolysis. Regardless of whether this is stress or shock, the pollen and ovule are always affected. This causes infertility in the pollen and ovule as a result of accelerated senescence induced by salt. It is difficult to study these structures and how they are impacted by salinity because they're hidden within the flowers of a plant. To understand how gametophytes respond better to stress or shock, the gametophytes of ferns were used. The fern used in this project was Ceratopteris richardii. The decision to utilize this fern is due to the fact that Ceratopteris develops from spores that can grow into a two-dimensional flat sheet of cells, making it easy to understand the effect of salt stress since no tissue manipulation is required. Additionally, this fern, unlike many other ferns, has a sequenced genome. To identify genes ubiquitously expressed in salt stress and shock, I used a publicly available RNA-seq dataset that contained gene expression data from the seed plant Arabidopsis thaliana, as seen here. I used integrated differential expression and pathway analysis to normalize the data. After normalizing the data, a principal component analysis was generated, showing distinct separation between groups. To select genes that were definitively regulated, I used a cutoff value of greater or less than a twofold increase with a p-value of less than 0.5. This allowed me to narrow down the dataset to 77 genes that were the most significantly regulated by salt. Analysis of these genes showed that the top-down regulated genes are members of the phytohormone ABA signaling pathway that includes an ABA receptor PYL6, as well as a PBTC, an enzyme that interacts with ABA receptors, as well as an LEA. Other proteins expressed were not highly characterized in plants, with the exception of MKK7. The only two genes upregulated were WSD1 and RHS16. Among these genes, MKK7 was the most conserved, and studies have shown that there's a correlation between increased MKK7 expression and salt tolerance in Arabidopsis thaliana. Several studies have shown that this protein is highly conserved. To see if this regulation is conserved in seedless plants, I used Ceratopteris richardii. To identify an ortholog of MKK7 in Ceratopteris richardii, basic local alignment search tool analysis was performed as seen here. At the protein level, there is approximately a 56% identity to various MKK7 genes in other plants. This MKK7, however, is more closely related to those in seedless plants than those found in seed plants. This gene evolved before the separation of seed from seedless plants, but is more similar to seedless plants. Analysis of structural features indicates that MKK7 has several of the essential motifs conserved, suggesting that it would be a functional protein in Ceratopterus ricardi. This includes known residues that are phosphorylated, such as these serine residues. To determine how salt affects gametophyte development in Ceratopterus ricardi, plants were grown in high levels of NaCl as seen here. A high level of NaCl reduces cell number and chlorophyll production, as well as development of the gametophytes. To determine if MKK7 was affected by salt stress, an RT-PCR was performed. As seen, a high level of NaCl reduced MKK7 expression. In Ceratopterus ricardi, there are many available mutants that show tolerance to high salt. I screened several of these mutants and identified one labeled STL1, which had a high cell division and chlorophyll production when grown in salt, in contrast to the wild type. This mutant also displayed a high level of MKK7. This data is consistent with previous studies that have shown an increase in MKK7 in Arabidopsis thaliana and Oryza sativa can improve salt tolerance. The similar downregulation of MKK7 in Ceratopterus ricardi as upregulation in the salt-tolerant mutant indicates it would be a good strategy to use in seed and seedless plants. Future studies would involve characterizing the effect of salt tolerance on Ceratopterus ricardi, as well as upregulating MKK7 in wild-type gametophytes to determine if the effect of MKK7 on salt tolerance is consistent with those seen in the salt-tolerant mutant of Ceratopterus ricardi. In conclusion, I found that MKK7 is a good candidate for improving salt tolerance in plants exposed to high salinity. This was observed due to the upregulation of this gene in a salt-tolerant mutant of Ceratopterus ricardi when it was exposed to the salt shock condition. Thank you for listening to my presentation.